start. And yep, there it goes. No, it's not the last session. Just let me quickly go to share screen. Start broadcast. 1, 2, 3. And we are in business, aren't we? Can you see my screen? Yay. Fantastic. Excellent. And please don't forget to register your attendance. Oh, you are very sweet. Thank you. Yes, BI 300, we have another session tomorrow, and I want to use that as a sort of a mop-up and, all the things that I've forgotten to mention. But then you get rid of me, at least for BI 300. For BI 308, for the skills module, I will still stay with you guys, so don't worry. And next term, then we have a whole module together, which is enzymes and metabolism, which is a lot of fun, actually. So, yeah. Actually, for BI 300, we are still doing then something in week 12. If anyone is still then alive in week 12 and has not collapsed because of overload. Nah, you're not. Yes, you are going to have another program level multiple choice assessment in week 6. Are you aware of that? Okay, good. Because at the moment, I'm currently writing the questions. We don't have the week off. You must be joking. No, you don't have a week off. If I can't have a week off. No, you're not interested in a preview. You are not interested in the questions. Not the biomed engineering students. And in week 7, you only have labs. That's right. No lectures. Do you actually want to know the questions for the BS 300 for the multiple choice? Yes, I thought so. Can you keep a secret? Uh-huh. Apparently only Hassan can keep a secret. Anyway. So can I. But honestly, the questions are very, very easy. The questions will be very, very easy. And if you have worked a little bit on this module, then you should not have any problems. So let's get started. Mean? No, I'm average, not mean. Anyway, let's get started. So just a quick recap. Um, about we've done the last couple of times, we started with amino acids and continued then with protein structure. And I told you when you are revising, you need to, let me write this down. You need to know the structure of amino acids, structure of the amino acids, but I don't ask you to draw them, so don't worry about that. But you need to recognize the amino acids. You also need to know the properties of the amino acids, and you need to know the one-letter abbreviation. Abbreviation of the amino acids. That is what I expect, okay? Properties, some people have contacted me and said, oh, we are not entirely sure about the properties. For example, there's the amino acid tyrosine. Does anyone remember the one-letter abbreviation for tyrosine? Absolutely, that's the Y. And the rest, the R group of tyrosine is basically CH2, and then we have an aromatic ring with an OH group at the end, right? This is what tyrosine looks like. And some people said, I'm confused. Tyrosine turns up as an polar amino acid and also as an unpolar amino acid. What is it? Well, tyrosine is an amino acid. It can be, in the right environment, it can be pretty much unpolar because of the aromatic ring, which is very hydrophobic, but it can also be polar if you look at the OH group. It's bipolar. Yes, I like that. That's a good one. But at least this is an aromatic amino acid. Yes, you do need to know the different R groups for the amino acids. But as I said, you don't need to draw them. No, it's not a funny joke. I absolutely agree with you. But for tyrosine, it has both properties. The protein's full name, I would try to avoid that. Another thing is we have 3 aromatic amino acids. So we have tyrosine. Writing goes down the pants. What is the other amino acids? It's F. Yep. That stands for phenylalanine. Yep. And the third one? W. That stands for tryptophan. Exactly. And they all have aromatic rings, right? And now here comes a big revelation. Aromatic rings absorb light, don't they? We had that with DNA. I told you, DNA absorbs light at 260 nanometers because of the aromatic ring. And so do amino acids that have an aromatic ring. So these three aromatic acids, YFW, which have aromatic rings, in a spectrophotometer will absorb light, but they don't well, they do absorb a little bit at 260 nanometers, but they predominantly absorb light at 280 nanometers. Absorbance. Because of the delocalized electrons. And do you remember when you did when you looked at the purity of DNA, you did the ratio of 260 to 280 absorbance. And at 260, we said that's the DNA absorbance. And now you know why we measure at 280, because that is where the aromatic amino acids in proteins absorb. So that's the protein absorbance. And that is the reason why we do this ratio, the 260 to 280 ratio. Because tyrosine, phenylalanine, and tryptophan, all three absorb at 280 nanometers. So that's the amino acid story. Now, I told you that we are looking at protein structure. And we said we've got the primary structure that is determined by the genetic code. We then have the secondary structure with alpha helix, and some turns, and yeah, beta turns, loops, and things like that. But they are predominantly kept together by hydrogen bonds. These are the forces that keep these things together. And we find that these protein structures, these secondary structures, will form higher order structure, the tertiary structure, where we have all sorts of interactions with between the R groups. So, how do I write this? Interactions between R groups, and things like that. And this then gives a protein its characteristic shape. And of course, and you get the disulfide bonds, and you get the ionic bonds in it. So all these structural elements that keep the tertiary structure together. And you find then certain amino acids, of course, being sort of either in the hydrophobic core or on the outside. So which amino acids would you find in the hydrophobic core? What do you think? Which amino acids would be in the hydrophobic core? Inside, protected from the water environment. Remember, the water was bullying the hydrophobic lads. Alanine, for example. Yes. What else? Non-polar aromatic amino acids. So hydrophobic amino acids would be in the core. Amino acids in core. And on the other hand, on the outside, you would, well, glycine is a sort of, is a weird one. Glycine can be basically everywhere. Isoleucine, yeah, that's a good one. That's in the hydrophobic core very often. On the outside, you would find, for example, charged amino acids. Very often you'll find polar amino acids on the outside. So, And when we talk about a hydrophobic core, this is the hydrophobic core. That's the interior of the protein that we are looking at. So hydrophobic amino acids in the hydrophobic core, hydrophilic amino acids, polar amino acids, charged amino acids on the outside of the protein, on the exterior. Okay, makes sense? A little bit, and that this is actually a very, very interesting, very topical subject. So protein folding. And this actually deals with how a protein goes from the primary structure over the secondary structure. I'll do it like that, where we form the hydrogen bonds, form the helices, form the beta sheets, and these local interactions to the native tertiary, and then also, if we have several protein chains together, the quaternary structure. So we have this process going from the amino acid sequence in the genetic code to the final structure. And we don't totally know the rules. We don't know exactly what dictates from the amino acid sequence to get to the native in. We know that the code that dictates the folding and what the protein actually looks like, we know it must be in the primary structure. But we don't know exactly how this code looks like, what the rules of this code are. What are the rules of the code, of the folding code? Let's call it that way, of the folding code. What are these rules? And we don't know it at the moment. And here is something that you probably have been waiting for a long time. So we have no idea, well, we have only very limited idea how this, the genetic code, basically, the amino acid sequence, how that actually dictates what the native protein eventually looks like. And I just quickly go over to this one here, because I want to show you something. Actually, wait a moment, I need to go back. Something that you are probably, that you're not expecting. Where's my genetic code? Okay, yes, let's add this. Ooh, wow. Okay, so here we've got the genetic code. Isn't it the R groups that determine? Yes, absolutely. But remember, we've got hundreds of R groups. And they all form then a stable form of the protein. So this is our genetic code. And let's look at the code CUG, right? What does CUG, which amino acid does it code for? Okay, that's the amino acid leucine. So that's an L, right? So L. And let's do CUA. What does CUA code for? Also leucine. Absolutely. So that is when people say the genetic code is degenerated. Because the same amino acid is encoded by two different codons. And that is what you learned, that the genetic code is degenerated. Now, a few years ago, people did a funny experiment. What they've done is they took a gene, I can't remember exactly what gene it was, and replaced the CUG codon, or in the DNA, it would be CTG, because we replace U with T in DNA. They replaced, and the protein with CTG in it was absolutely nicely folded. So that's the protein. Was a nicely folded protein. Okay. Now they replaced it with CTA. Would you expect that it made any difference in terms of amino acid? What would we get if we replace CTG with CTA? Would we expect what do we get if we replace CTG with CTA? We still get the same thing. So we should get the same outcome. And yet, when they used CTG, they had a nicely folded protein. When they used CTA, they just got a mess. So the substitution is actually, we substitute for the same thing, but something fundamentally changes. We change a leucine for a leucine. And the protein is totally messed up. And why could that be? If you look very carefully, you find that for some degenerated amino acids, there is what is called a codon bias. So people have done a lot of bioinformatics. Yeah, Mohammed, exactly. They changed one codon. Actually, they changed one base from a G to a A, and it has a dramatic effect. And it didn't even change the amino acid. They said nothing changed. A-level slide. There's a codon bias. So CTG in humans is used 47% of the time when you want a leucine, whereas CTA is used only 7% of the time. It has nothing to do with the basis. It has something to do with The DNA is transcribed into the mRNA, and the mRNA is then translated into a protein. Now, CTG is used very often, so you've got lots of CUG tRNA, if you like, which delivers the leucine. But you have less of the CUA tRNA because you don't need it so often. It still delivers the same amino acid, but because you have less of it, the protein gets a chance probably to misfold. Absolutely. And while it is fully understandable if you, for example, change a leucine, a hydrophobic amino acid, to, let's say, a lysine, which is a charged amino acid, that you mess up the protein, even if you change the codon and have the same amino acid in place, you sometimes end up with a total mess. Mohammed, you absolutely nailed it. So for everybody who doesn't see the chart, if you have a mutation that still gives you the same amino acid, yet what could happen is that the protein might not adopt its correct functional state. I think that's absolutely amazing. Are you amazed? And especially that A levels lied to you. So the genetic code is neither universal, nor is it totally degenerate, because my personal view is that each of these different codons, actually has a function, and the cell can, for example, measure how much of a tRNA is present, and therefore can adjust protein folding to the needs. Now, I told you we don't know the code that rules it. You probably will be told. Life is an illusion, and I'm a magician. We the genetic of protein folding, and we do not know, or we can not really predict what the structure of the the native protein will look like. So, because we don't know what the rules are from the primary structure to the tertiary structure, we do not know what the proteins look like just by looking at the amino acid sequence. However, we are working on that, and we do a lot with bioinformatics. Oops, hang on. Am I back? Yeah, I should be back. We do a lot with bioinformatics. Because if we find out the structure of a protein, structure of one protein, we can then say we have other proteins that have a similar amino acid sequence, And therefore, we assume that similar proteins have a similar structure. So with bioinformatics, we can do that. And we just simply say, yes, we've got a structure. We look at other proteins. We have a similar amino acid sequence, and therefore we assume we have a similar structure in these other proteins as well. There also have been people, very clever people, for example, Chu and Fassman, who just looked at secondary structures of lots of proteins and just did a count of amino acid distributions in secondary structures. And they found, for example, that an alpha helix has fewer glycine and proline residues in them. They counted a lot of amino acids, and with a computer program, you can easily do that. So what you can do is you can simply run a Trufastman program, and my colleague, Dr. Mark Was, will tell you more about that. And this Trufastman program will then look at the amino acid sequence and say, oh, look, we've got here a stretch of amino acids where there are hardly any G's or P's in it. Oh, look at that. No G's or P's. So if there's no G or P, then it is likely that we have an alpha helix here. And there are lots of prediction programs now. No, what study? Replacing the synonymous codon. Are you referring to that, Evie? The alpha helix. Well, it is on the slides on Moodle from my colleague. So you will find that alpha helices usually don't have a lot of G or P's in it. It's not true that they don't have any G's or P's in it. There are proteins with helix that have G or P's, but they are sort of general rules. Oh, the Trufastman. I mean, it's probably not so much known under that name. It is just people say, oh, well, you've got less glycine and proline there, so it's likely you might have an alpha helix there. Or they say G and P are what is called helix breakers. So glycine and proline are helix breakers. But we are trying to work, and with increased computer power. Hang on. Why does it do that? With increased computer power, we can do more and more analysis of existing proteins and can look at what these proteins actually look like. So how can we actually analyze proteins? And one of the helix breakers, they are called helix breakers because they would very often interrupt a helix, and that's why they are not found in a helix. So analyze proteins. Now, we discussed how we can analyze DNA, and we said DNA can be easily analyzed if we are looking at different sizes of DNA, size of fragments. We can subject the DNA to an electric field. So, electric field. And in the electric field, if we have it negative to positive, if we start our DNA here, so that's the start point, the DNA and the DNA would move from negative to positive. Why does the DNA move from negative to positive? DNA. Why does DNA move from negative to positive? It's negatively charged. Exactly. So DNA is negatively charged and therefore moves in that direction. And then we said we use a solid support, and we use this compound agarose that is sort of a mesh where the DNA molecules have to move through, and we can order them by size. So large molecules, large DNA molecules would move slower, smaller molecules would run faster. Okay? And the phosphate gives the DNA its negative charge. Now, can we do something like that with a protein? And the answer is, in principle, yes. But we have a number of problems with proteins. What is the charge of a protein? Positive, neutral. Oh, wait. Charlotte, absolutely right. It depends on the amino acids. So we cannot say a protein will be positive or negative or neutral. It really depends on the amino acid. So charge of a protein depends on the amino acids. Absolutely right. Now, with DNA, we also had the simple situation where each DNA molecule had the same, if you like, structure. The DNA was just simply the double helix. And it basically had the same size in a way, the same shape, I should say, same shape. Okay? Unfortunately, we don't have this for proteins. Proteins come in all sorts of different shapes. Which is a real pain. Because imagine you have a sort of a mesh like agarose, where you have pores. Now, you might have a protein that is a big protein, or you have a protein that is really densely packed, is just totally squished together. They both might have the same amount of amino acids, but they have very different sizes, different shapes, if you like. They have the same length, same number of amino acids, but very different sizes. So how can we analyze proteins? Well, what we want to do is we want to separate them like DNA, separate them by size. Like DNA, so that we have that we have proteins, and the smaller the proteins, the faster they would run. But how do we actually make them run? The trick is we open the proteins, open proteins, and open, that is sort of, I put that into quotation marks. We denature the proteins. Denature. Denature in this case means we destroy the native structure. Destroy native structure. So then all shapes will be sort of unraveled. And the shape does no longer make any difference. How can we denature proteins? Does anyone know? How can you destroy a protein? Intense heat, exactly. You heat them up. Heat up the protein. Can also do that with acid, but heat is much easier. You heat up the protein. So you have a nice structure like that, yes? You heat it up. The molecules in this protein, the atoms begin to move around because it's getting warmer. They move around, they're getting even more warm. Move around, move around, move around, move around. The whole structure collapses at high temperature. What will happen when you have opened up your protein? What happens? Remember, we have hydrophobic patches here. What do they do? These hydrophobic patches. Come back to that in a minute. The hydrophobic patches would stick together, wouldn't they? Remember, the hydrophobic lads get bullied by the water. They will stick together. They will run away. So if you heat up a protein, you will destroy the native conformation, but the whole thing will clump together. You have experienced that. You have experienced that before. When? Anyone? Eggs. Explain. You're right. I like the idea of the eggs. Explain. What do you say with the eggs? When you boil an egg, or when you fry the egg, when you fry an egg in a pan, what you do is you apply heat. The egg white that is there in the native form becomes denatured. The hydrophobic patches that are usually buried in the hydrophobic core of the protein will be exposed and clump together. And that is how you make an omelette, exactly. So what we need is we need to avoid these things sticking together. And ideally, we also want to not worry about the charge of it. So what we do is we heat up our protein in the presence of a compound that is called sodium dodecyl sulfate. And I've just done an exam question about, oh, sorry, I should not have said that. Damn it. Forget that, okay? So let's write this properly. Sodium dodecyl sulfate. And this is a very hydrophilic compound with CH groups. And at the end, we have an SO3 minus, and we've got sodium as well there. Now, the important thing is that this, it's also called SDS, student data system. These molecules of sodium dodecyl sulfate will actually
not my friend, sorry. One of the they were talking about it, and they said that their kid, they had to buy it online, so that's why they didn't get it on the laptop.
Yeah, I fully agree.
What is that? Mum's phone. Mum's phone. It's so low. Yeah. I don't get what's happening. Yeah.
Yeah, our attendance gets younger and younger. That's good. I mean, catch them young. So we have this protein that has been sort of exposed. It has been opened up. It has been denatured. And now what we do is we stick these SDS molecules into it. And because of this hydrophobic tail, it will stick to lots of these to the protein. Now you can see it has this hydrophobic tail that sticks to the protein, but at the same time it also has the negatively charge on the outside of the sulfate group. So our protein will be negatively charged. So if you have another protein sitting next to it, again, with this SDS sticking out, will they clump together? Do these proteins clump together now, like when you fry the egg? Absolutely not. This SDS prevents the proteins, the denatured proteins, from clumping together. And it also makes them uniformly negatively charged, like our DNA molecule. So all we need to do really is to use this SDS and boil our egg in SDS. This is actually it's like a detergent, but a very strong detergent. So if you boil an egg in SDS, it will not clump together. You will not get a fried egg. And what you can do now is you can use this heated sample, heated plus SDS protein sample, and load it on a gel like the agarose gel. Fluff's sake, why is it cutting out? Apologies for that. Yeah. So you heat your protein in the presence of SDS and it becomes negatively charged. And now we can run this like our DNA on a gel. And we run it from negative to positive. And now we can separate the proteins by size. So the long proteins with large number of amino acids will run slower. The shorter proteins will run much faster. Does that make sense? And we have, because they are all negatively charged, we've got negative charges on all the proteins, and the bigger they are, the slower they get through the matrix of the gel. If they are very small, they move faster. If they are very big, they move slower. But unfortunately, it's not that easy. Now the SDS binds to the protein because SDS has this very hydrophobic region here. That's lots of CH groups here, exactly 12. It's very hydrophobic, and therefore it interacts with the hydrophobic bits on the protein molecule. Okay, that's how this SDS sticks to the protein, to the denatured protein. You will find loads and loads of videos on YouTube about protein electrophoresis. And so have a look at protein electrophoresis. Or sometimes it's also called SDS electrophoresis. Bottom line is, if you heat the protein in the presence of SDS, you fold it up and or you open the protein, and then you can separate proteins just simply on the basis of their length, of the size of their amino acid, or of the number of the amino acids. So this might have here 200 amino acids. Maybe this has 150 amino acids, 175, 20 amino acids, something like that. Because SDS makes everything negative, you have the similar situation to DNA. DNA is all negative, so we don't have to worry about making it negative. But because proteins can be positive or neutral or negative, we have to make sure that everybody is sort of made equal, and all the proteins are then charged negatively so that we can run them in this direction. We will do a little bit more about the SDS electrophoresis tomorrow, but ideally what I want you to have, what I want you to do is check out YouTube and see if you can come across some good videos which you can email me the link to so that I can put it up on Moodle. Obviously, I will acknowledge that you found it, so I can say, John Smith found this great video. All have a look at it. So thank you very much for watching, and I will see you tomorrow afternoon for the BI308, and then late afternoon for the last installment of proteins. So thank you very much, and have a lovely afternoon stroke evening. I'm ready for my afternoon nap. Are you? Take care. Bye-bye.